Hi 232 students, welcome back to Human Anatomy and Physiology Lab at Clackamas Community College. I'm Christina. Today we're going to talk about exercise 29, which is blood. And so we're going to look at all the formed elements in the blood, including red blood cells, white blood cells, and platelets or thrombocytes. We're going to do a white blood cell count differential. So you're going to basically be kind of figuring out percentages of white blood cells in a smear. We're going to talk about blood tests, so different types of things that they test blood for and why they would do those tests. We're going to talk about a few blood disorders, and then how to type blood and where your blood type comes from. So here we are at your lab report. The first part of your lab report just has you draw or make a note of all the different types of cells. So remember, you do not have to upload any histology pictures to your lab reports. I would urge you to draw them at home, or just have a couple bullet points that tells you one type of cell apart from another. And then the other part of knowing which cell is which is what their functions are. So what is the specific job a neutrophil does? What is the specific job an eosinophil does? So I'm going to go through each type of white blood cell for you that you'll be looking at histologyguide.com and identifying cells. They do a great job of pointing them out to you. First thing I want to point out is that these three look a little bit different than the other three, or the other two, apart from these two. So these three that I first circled are granular, and that means that they have vesicles of product, a hormone or an enzyme, that they carry or move to their cell membrane and that they release at an injury site. Two are agranular, like amoral, without morality. And they don't have any vesicles on their cell surface. The cell itself is doing the job. So I'm putting these in my very own order, or not the order that they are on the slide. I'm not going A through E. I'm going in order of this mnemonic, never let monkeys eat bananas, because it tells you the order of prevalence of neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So we're going to talk about neutrophils first. Neutrophils have a multi-lobed nucleus that looks like a string of small sausages. And so you can kind of see all these connected lobes in there. They're covered in granules, so they have vesicles on their surface. So there's going to be some dark blue polka dots on them. And those vesicles are filled with product that they release. They're the most abundant of all the white blood cells, and so if you are uncertain about what cell you have seen, it's likely a neutrophil. And their overall job is to phagocytize pathogens and kill bacteria. Next cell that we're going to look at is a lymphocyte. Never let monkeys eat bananas. And lymphocytes are indicated, they have a dark blue, almost spherical nucleus. Sometimes it can have a little indent in it right here. And then their cytoplasm extends kind of outward in this little crescent moon shape right there. And so that's my memory trick for a lymphocyte. It has this little crescent moon of cytoplasm, and so I think Luna lymphocyte. That's my memory trick for that. And they are agranular, so no vesicles, and their main job is to fight viruses and cancer. They're kind of a general immune responder. Cell is a monocyte, and monocytes are monsters because they're the biggest white blood cell, and they have a kidney-shaped or horseshoe-shaped nucleus. And so we can kind of see that horseshoe shape or kidney shape right there, with a large amount of kind of grayish cytoplasm on the outside of it. And their main job is to phagocytize pathogens and debris, and they literally go around engulfing a bacteria or engulfing a piece of debris and ingesting it, therefore killing it. Pretty dang cool. That's why they're the monster. Next are eosinophils, my very favorite cells, because they have hot pink to orange granules on them, and they're just very pretty. So they have a bilobed nucleus, and so you can kind of see that bilobed nucleus right here. And then they're covered in a bunch of polka dots, pink or orangey polka dots everywhere. And that's your big tell for an eosinophil. And their main job is they are heightened in allergic reactions, and they kill parasites, parasitic worms, and stuff like that. Last white blood cell is a basophil. Basophils are very elusive. They're the least common white blood cell, and it kind of can be like finding a needle in a haystack because you'll look all over your slide and you'll sometimes barely find one. They have a lobed nucleus, although it's not often visible. I can kind of outline the nucleus right here. Sometimes you won't see it. It'll just like a big dark blob of polka dots. They have tons of vesicles on their surface, so all of these dark black to blue polka dots all over them are vesicles of histamine and heparin. And so that's their job. They are active during inflammation, and they come to an injury site and release their vesicles of histamine and heparin. Next thing you're going to do on your lab report is a differential white blood cell count. And there are instructions in the online instructions that tell you how to do this, and they send you to the first link on the slide, which is the peripheral blood smear, which I will show you in just a minute. Here is the peripheral blood slide. And so what you're going to do, first I'll make note for you that you can see all the different types of cells over here. So just click on the thing you want and it'll zoom right in on them for you. And what we're going to do, I'm going to go across that slide, just kind of counting across, and you're going to make a tick mark in the box for every neutrophil you see in that row and every eosinophil you see in that row. And then you're going to kind of go down and then across again, just making tick marks every time you see. Maybe you'll find the one lone basophil on your slide in that row. And then eventually you'll get a total. So you'll get a total amount of neutrophils that you count, and it'll be divided by the total number of cells on the slide. So Michael Patterson says there's roughly 60 to 80 white blood cells on this slide. If you count to 50, I'll be happy. After you fill out your values from your differential count up here, you're going to answer some questions about typical diseases and disorders of the blood that we might be indicated by those tests. All right, so you have about 6 different diseases here that you have to talk about. One hint is look at the words, just like knowing how the muscle names tell you a little bit about where that muscle is. You might see some vocabulary in here that indicates specific things. So if you're looking for the cytosis as a suffix, we have a couple of them here, leukocytosis and thrombocytosis. And so we can infer a little bit about the type of disease these two are by that word. Cytosis means an elevation of cells in some way. And I've given you a beautiful example of a sickled cell right there. The next part of your lab report has you looking at different tests that you would do. So I'm going to do the first one for you. The first test was a total white blood cell count. So these are the basic things you have to know about each test, or tell me about each test. What is this test? And I don't want to know how it's done. I don't want to have anything about, oh, they centrifuge the blood and they separate this out. Nothing like that. A total white blood cell count is a measure of the total number of leukocytes per unit volume of blood. So we're looking for elevation or decreased levels. And so I have to know what normal white blood cell values are in order to tell if they've been elevated or decreased, and then what conditions would cause an abnormal test. And so an increase of white blood cells comes oftentimes from an infection of some sort, bacterial or viral, and if I have an increase in white blood cells, it's general leukocytosis, and if I have a decrease in white blood cells, in general, it's leukopenia, and I would know those ranges. So these are the rest of the tests that you have to know. So you're going to do a total red blood cell count. You're going to do, and why, why would you look at the amount of red blood cells somebody has? A hematocrit, which has to do with packed red blood cells versus the amount of plasma you have. Hemoglobin concentration has to do with the ability of a red blood cell to carry oxygen. Sedimentation rate, coagulation time. And then you have some stuff to do with cholesterol. And so you're going to define some terms here and give a normal cholesterol range. And then within that normal cholesterol range, you're going to dig a little bit more and look at the difference between high density lipoproteins and low density lipoproteins, and you're going to tell me why it's good, why I can have tons of the HDLs and I do not want to have tons of the LDLs. And the very last part of the lab is all about blood typing. So Professor Patterson has posted a video for you to watch in the online instructions. And then the last person that they test in the video, you're going to record those values right here. That you're basically going to be figuring out what blood type the last person in the video has. And here in the lab instructions is all the information about blood typing and the goofy but accurate video that you have to watch. I tried to click on this blood typing game. It might work for you. It didn't work for me, but try it. So here we are. I just want to kind of go over how we come up with blood type. And so these slides right over here, you're going to be seeing in the video. And so I'll talk about that in just a second. But first off, I want to talk about how you get your blood type. There are two major differences in the factors that give us blood type. One is the antigen, and antigens live on your red blood cells. And the second are antibodies, and antibodies, I put it in yellow to remind you that they float around in the plasma, and plasma is kind of a yellow color. So antibodies live in the plasma, antigens live on your red blood cell. So let's start with a type O person. A type O person doesn't have any antigens on their red blood cells. Do you see there's no structures on the red blood cells? But they do have lots of things floating or able to be floating around in their plasma. And so here are all the antibodies that can float around in their plasma. And they're called anti-A antibodies and anti-B antibodies. So an anti-A antibody has this funky kind of purple triangle shape in this diagram right here, and it's going to clump with any red blood cell that has a little A antigen on its surface. And so here we have an A antigen on the surface of a red blood cell. And so if you put this little triangular thing right here with this antibody in the yellow, they're going to clump together, and that person is going to die because all their little capillaries are going to be filled with weird clumpy looking stuff, and oxygen can't get to cells, and that's bad. So a person with type A blood has A antigens on their red blood cells, and they will generally have anti-B antibodies floating in their plasma because they're just waiting for any B-flavored red blood cells with B antigens on them to show up. They're just ready to attack them and clump them so that they don't invade the rest of your body. A type B person has a B antigen on its blood, so the little circles right here, and it's going to have anti-A antibodies floating around in that person's plasma, ready for those foreign A-flavored red blood cells to show up so they can kill them. And then a type AB person, new color, a type AB person has both an A and a B on their antigen on their red blood cell, and so they can't have any at all antibodies floating around in their plasma or else it would kill them. So no antibodies in their plasma, but 2 flavors of antigens on their red blood cells. So, in the clumping test that you watched a video of, I'm going to put this blue colored kind of anti-A antibody on a drop of blood. And because I see clumping there, that means that this drop of blood had little A's on their red blood cells, and then they clumped together. This drop over here got anti-B antibodies put on it. And because we have all this clumping, that means that that person also has a B antigen on their red blood cell. So that means that person is type AB. The positive and negative comes from RH, and RH is just another flavor of antigen that can live on your red blood cell. So you either have one and therefore you're positive, and you won't have any antibody floating around in your plasma. And if you don't have one, it means that you might have antibodies for anti-RH floating around in your plasma, but probably not. You have to be exposed to it to have an antibody. So this next person, we put anti-A on them and they clumped, so they do have an A, but we put anti-B antibodies on that drop of blood and there was no clumping, so there's no type B there. So this person is a type A. This one right here, we put anti-A antibodies on the blood. We got no clumping, so there are no A antigens in this sample. But we did put anti-B antibodies on, and we got clumping, so there is type B person. And then this last square, we put anti-A, no clumping. We put anti-B, no clumping. So that means there are no antigens on those red blood cells. That means this person is type O. And that's going to do it for exercise 29. I'll have a new video for you next week. Thanks.